Okay, so we're going to draw. You know I'm so good at drawing, so just be prepared. We have never seen anything that looks this good. So you've actually taken notes on all 4 of the macromolecules. There's a little bit more information that we need to do on DNA and RNA, but it's honestly, if I was teaching this, this is exactly how I do it. We do it in its own little video. So you're going to want to go to your next blank page and see how I didn't use this one, because I want to have the ones across from each other. So I'm going to just put, DNA here, and we're going to draw it. So I'm just going to tell myself this is what it looks like. And I'm going to give you the way I draw, and then we'll see what you think. So the way that the sugar looks, which is the directionality of the molecule, it looks like a little house with a chimney. I mean, that's just the way it actually looks. So the way I draw it, I just repeat this, is I usually draw a straight line like this. Okay, so notice I'm using like 4. You can do whatever you want, but it just gives me some kind of proportionality. Then I'm going to make this look like a house with a roof and the biggest chimney ever. I don't know why it's so tall, but that's what it looks like. So this is your sugar, and carbons are going to be named based on the position where they are. So we're going to call this one, this is your 1 prime, and then you just walk around and you number the rest of them. So down here is the 2 prime carbon. This is your 3 prime carbon, and then it gets a little weird. Your 4 prime carbon is here. Okay, well, I see that. That makes sense. Those are all the little vertices, the little corners. I get it. But your 5 prime carbon is not here. This is actually an oxygen. Your 5 prime carbon is up here at the top of the chimney. Okay? So there's 1 sugar. Your DNA we talked about, sugar, phosphate, sugar, phosphate, sugar, phosphate, right? So here's the sugar. I'm going to connect the phosphate group is between the 3 prime sugar of 1 sugar and the 5 prime end of the next one. So I'm going to have a phosphate group. Let's put it here. And it's going to be covalently bound to this 3 prime end of this sugar. And then I'm going to draw my next sugar. So I'm going to just do the same thing I did before. So I'm going to go down through here. I'm going to make my chimney, make the base of my house. I'm going to go ahead and put my oxygen at the top. I'm going to number. So that's my 1 prime, 2, 3, 4, and the weird one up here on the top of the chimney. And that's where the phosphate binds to the next sugar. Repeat. So I've learned, first of all, the sound quality is crappy. So I ordered, I don't know how it's going to do, ordered this cool little microphone thingy. You probably can't see it. This thingy. I'm going to try it in class, and I'll try it in here. We'll see if it makes the video sound better. I think it's because this room is so empty without you. Anyway, so the sound's terrible, but we'll work on that. And then I know that things get really noisy because I think the sound comes through this guy. That's probably really loud where I tapped on it. So when I move it, that's what's so awful. But I have to move it because then, you can't see what's happening. So I'm going to try to be a little better. Look at that. Did that all by myself. So I'm going to repeat this again. So I'm going to have, this is my sugar, my phosphate, my sugar. It's time for a phosphate. Put it right here. And then it's going to bind to the 3' carbon of the one above it. Then I'll draw my next sugar. Just repeating, love repeating patterns. They're all in nature. I love them. I'm going to rename all of my carbons. 1', 2', 3', 4', 5'. So the phosphate is bound to the 5' of the one below it. Repeat last time. So I'll put a phosphate group here and my last sugar. So I'm going to draw this little guy and number 1, 2, 3, 4. And up here is my 5. The phosphate is bound covalently to the 3' of one and the 5' of the other. We'll come back and label those in just a second. All right. So there's the directionality. You can see, I think I told you, I know I did, I typed it, when the woman said that they were oriented in opposite ways, anti-parallel. Notice that all of the sugars are pointing in this direction. So this is the 5' end. Makes sense because that's where that it ends at the 5' sugar carbon, 5' carbon of that sugar. And then down here is the 3' end. So I'm going to write that. This is the 3' end. Okay, you could probably want to see that. And then right here is the 5' end. So then what I'm going to do is I'm going to take everything I drew this way, it's like an interstate, and I'm going to drive it back in this direction, which means all of my pointing in this direction sugars are going to point in this direction. And it helps if you stagger the sugars, only because of the bonds we make in the middle with the nitrogenous bases. Trust me on this. So I'm going to go up here and I'm going to draw the same thing with my house. So, except upside down, right? So there's my chimney. And I'm going to have that same oxygen there. And then I usually, just because it's easier for my brain, I'm going to just number them in reverse order, starting with this chimney and just walking around. And then I've got that. Make sense? So then I'm going to have a phosphate group. And I've got room, I'll just put group right here. And then I draw my next sugar oriented in the exact same way. So I'll do the same thing. Now let's be truthful. That's pretty good looking. 5', and I'm just walking around. Now if I put the 1. Phosphate group, covalently bound to the 5' of one and the 3' of the next sugar. Here we go again. Put a phosphate group. Sugar, phosphate, sugar, phosphate. And then I'm going to just draw, whoops, I kind of got off on my little thing there. So there's my next sugar. I feel like they're moving in. Maybe they're not. I'm really bad about like running off the page. So I'm just going to put what prime carbon each one of these would be. Okay, I wanted to tell you a little secret. I'm like super renegade today. I didn't put my sign out that said don't come in here when I'm videoing. Not that it keeps people from coming in here. Every time I'm like, hey, wait just a second. That's because somebody walks right in. But I don't have it out there. Makes me a little nervous. So, covalent bonding, 5' end of this one, 3' end of that one. One more time, right? 1, 2, 3, 4. 1, 2, 3, here we go. Phosphate group, I'm not going to write group because I did put it up there, so I know what I'm doing. And I'm going to draw my sugar with the directionality of the 5' carbon down. So that looks like that. And so 5', 4, 3, 2, 1. And again, I'm going to put the covalent bond, 5' end of one sugar. You'll never be asked to draw this, ever. But you need to see it in your head, which is why we draw it. I could show it to you, meh. I could show you a video, meh. I could talk and point, meh. Your brain stores things in different places. What you hear goes in one location, what you see goes in another location, what you talk about goes in another location, what you draw goes in another location. All of that gives you many places to pull from to create a complete picture and to actually get what you're talking about and what you're thinking about. You have many different cooperative places where that you've stored information. So let's think about this. So now since the 5' carbon is in this direction, this is the 5' end. Ooh, that makes sense, doesn't it? Because I learned anti-parallel, they're going to always have the 5' of one with the 3' of the other. I get it. I see why. And the 3' here. Okay? So this is the way that we think about information flowing, is we have to think about whether we're at the 3' or the 5' end. So I'm just going to make a line here to make the point that this is all the DNA strand on this side of the double helix, and the information is 3' and 5'. All right, so a nucleotide, that's the monomer. A sugar, a phosphate, a nitrogenous base. I don't have that on here. Where is that going to go? Actually, those nitrogenous bases are covalently bound to the sugar phosphate piece, so you get your nucleotide at the 1' carbon. So all I'm going to do is just draw the bond out here, and I'm going to write that I would have a nitrogenous, am I going to have room? Yes, base, ATC or G. All of everything that I've drawn right now, covalent bond, covalent bond, covalent bond, super sturdy, will not come apart unless I purposefully do that. So I'm just going to repeat that at every 1' carbon on these sugars. So I'm going to have a covalent bond that has one of the 4 nitrogenous bases. So nitrogenous base, look how neat I'm writing. I'm going to do the same thing here. So I get to write nitrogenous base 8 times. 1' carbon, covalent bond, nitrogenous base. Not many people can say they've written nitrogenous base 8 times. Now you see why I staggered it. So covalent bond, right? Nitrogenous, why do I have room? I do. Base. We'll talk about this part here in the middle. Again, 1' carbon, covalent bond, nitrogenous base. The 1' carbon of the sugar, covalent bond, nitrogenous base. So much fun. 1' carbon, covalent bond, the last nitrogenous base. I'm so sad. That was so much fun. Okay, so now I'm going to take, purple sounds good to me, and I'm going to batch these into the monomers. Okay, so let's think about it. I'm going to have a problem. 1, 2, 3. I'm going to maybe put a phosphate. Let's put it down here. Or do you want to put it up there? Doesn't matter. Doesn't really matter. I'm going to put it up here. So, phosphate. And then, can you see? Okay, good. I forget to look sometimes to see. Okay. Sugar, phosphate, nitrogenous base. That is the monomer. That's one. Sugar, phosphate, nitrogenous base. That's another. Keep going. Sugar, phosphate, nitrogenous base. Sugar, phosphate, nitrogenous base. Nucleotide. That's going to be my nucleotide. Same thing over here. Sugar, phosphate, nitrogenous base. This is going to be wonky because of the... I'm like, seriously, look at that. I maybe have like, I don't know, 8 pages left. That's crazy. Again, sugar, phosphate, nitrogenous base. So I'm going to catch the phosphate and then just run it along the edge of the sugar. Catch the phosphate, run it along the edge of the sugar. Catch the phosphate, run it along the edge of the sugar. Good deal? This bond right here between each one of these nucleotides. So let's write on here, maybe down here, I've got room. So this is a nucleotide. Where'd my L go? Oh, don't be me. Nucleotide, right there. That's one. That's the monomer. It's weird when the basic part's made of 3 parts. Also, nucleotide. Let's see if I can not leave my L out this time. So I have 1, 2, 3, 4, 5, 6, 7, 8 nucleotides shown here. So this is my sad little small polymer. Okay, so far, this bond right here, this is a covalent bond. Goodness gracious. Covalent bond. So is this one. So is this one. So let's just write that on there. I think my pen's running out. I've gone through one already. Everything within the nucleotide and between the sugar phosphates of the backbone are covalent bonds. So between has a special name. This is what's called, I'm going to write it up here because I've got more room, the phosphodiester bond. And I'm going to highlight it in yellow. And it mushed it all. So all of the yellows are the phosphodiester bonds that are the actual name for the covalent bonds. All of these were created by dehydration synthesis. But the dehydration synthesis bond process made this covalent bond called a phosphodiester bond. It's between monomers. We've learned that over and over and over. So if this is DNA, let's see if I can manage. And this is DNA, right? Anti-parallel. What happens here? Like, what's this attraction between the nitrogenous bases? there are hydrogen bonds between, and there are either 2 or 3, depending on whether you're talking A's or T's or C's or G's. And so we'll just put those there. I should show you. So I'll put a little note here. I'll put hydrogen bonds. So they're easily broken, which means I could unzip this and zip it back and unzip it, because the information is right here. So you want to be able to get to the information, close it back up because it needs to stay shut, get to the information. So there's your hydrogen bonds. They're weak, they're easily broken. Okay, let me tell you which ones are which. So between adenine, remember Addie? A and T, there's 2, and it has to do with the position and how they meet up with each other. Yet again, the typical technology issue. My computer decided that it had no more memory, so I have no idea what I said last, but I'm going to just start from here. So the way that these associate is through hydrogen bonds, and there's either 2 or 3, and it has to do, I think this is where I was, with the way that they line up with each other, because they're not exactly the same. A is not the exact as G, and C is not the exact as, what did I say? T. Okay, so make sure I'm not crazy. But the way they line up, there's either 3 places where they're attracted or 2. So here's how this works. When you have the pairing of A and T, they have 2 hydrogen bonds between them. So it would be this one. When you have C with G, there's 3. So it would be this one. So let's kind of blow this part up and think about, okay, these pairings. What's the reason why that A only goes with T, not C? Let's see what the reasoning is. Or A never goes with G. So I'm going to just simplify this, and I'm going to put, I have a phosphate group covalently bound to a sugar. So this is the beginning of a nucleotide. And I'm going to, at this 1' carbon, I'm going to have a nitrogenous base, but I'm going to draw a very generic, just purine, something with 2 rings. So don't judge. There's my 2. And this is 1 nucleotide, right? So I'm just going to come down here and draw 1 more, but just do 1 ring, just for the point of what I'm making. A phosphate covalently bound to a sugar covalently bound to, this time I'm going to do pyrimidine, and this is another nucleotide. Let's covalently bond these 2 together, making that sugar phosphate backbone. Let me remind myself that this is the phosphodiester bond. And now I'm going to take this and I'm going to drive like down the interstate this way. I'm going to turn around and drive it this way. So I'll have the sugar to the phosphate, just because the orientation's anti-parallel. To the sugar is covalently bound. What does it have to be? If this is a purine, it has to be to a pyrimidine. So that's 1 nucleotide. So let's just draw a variation over here because I don't have a choice. It has to be a purine here. So I'm going to put sugar covalently bound to a phosphate group. The sugar is covalently bound to this purine. That becomes my 4th nucleotide. Let's bond these together. Phosphodiester bond. yellow. So I'm going to let this one be adenine, Addy. So that means this has to be thymine. And then I will let this purine be guanine and this be cytosine. So really I've just mimicked what's up here. So this would have 2, this would have 3. So what's the reasoning behind all this? What's the point of it? If you look, the C and the T, these pyrimidine nucleotides are smaller than the purine ones. But the width is standard here. It's always going to be 1 with 2, right? So this is the width. Or you could have 2, wow, that one went wonky, with 1, and that's the same width. We should agree on that. What's not the same, if you just said, come on, however you do, I don't care, hook up how you want to. What you would have is the possibility of a purine with a purine, which is too wide, or a pyrimidine with a pyrimidine, which is too narrow. Who cares? What does it matter? We care. We care. Because the way that DNA repair enzymes work is by width. So I'm going to let this be my DNA repair enzyme, and it's like train tracks. Right? And the width is correct because it's able to measure by width. So it would be able to measure here. So let it just be a cap. Perfect. Right? Here, nope. Here, nope. So it will catch the error made by you put an A with a G and you shouldn't have, or you put a C with a T and you shouldn't have. So this is the way that error checking is. So I'm going to make this in pink, and we're going to put that DNA error checking enzymes, molecules, are going to ride the DNA backbone. And what they're going to check for is they're checking for the correct, well, that got crazy, width, which you can only have with a purine with a pyrimidine. It's the only way it's going to work. And then the only way it's going to work here, if you think about this, if there's 2 hydrogen bonds, A cannot, because it's got 2, it can't go to C because it won't hydrogen bond correctly. So your hydrogen bonds here are going to ensure that A is with T because there's 2 places, and that C is with G because there's 3 places. So the width is about pyrimidine and purine only, but you could mess that up, right? Like you could have the wrong pyrimidine with the wrong purine. No, I cannot, because they won't combine correctly. Their association with these hydrogen bonds, they won't work. So it's sort of like, not exactly, but you know how I am. So here's this marker, and it's supposed to go with this lid. Okay, well here's the lid. What does it matter? Doesn't work, right? So you have to have the correct purine with the correct pyrimidine, or the hydrogen bonding won't be correct. And if you put them, you know, here, purine with purine, too wide, pyrimidine with pyrimidine, too narrow. So this all has kind of stupid proof involved, because it can only work together across here and with these two, correct width, purine pyrimidine, correct purine with pyrimidine based on the hydrogen bonds. Okay, so what I'm going to do is I'm going to stop this part right here, but I still need to walk you through some information about DNA and RNA. So, because of time, I'm going to bring that in as a different video, and I'll piece these all together. All right, we did some good drawing. All right, so this is where we left off. Drawn some amazing nucleotides, each one of the purple boxes, covalently bonded with a phosphodiester bond together along the backbone. And we have the nitrogenous base conversation about how they're hydrogen bonded, which makes it easy to unzip. And it's based on width, which is why you get one purine with a specific pyrimidine, because they have to pair up just right. So we want a place where we've summarized the differences in RNA and DNA. They're both nucleic acids. So I'm just going to give myself a little spot here. I'll make a little table. So there's what I'm going to put. So I'm just going to make myself a little table. It's not so great. And I'll have the ability to contrast. So this is the kind of thing that would be a really good short free response, if you think about it. You could be asked to contrast or compare and contrast DNA and RNA. So first of all, we'll start off with something simple. I think everybody immediately thinks, oh, okay, DNA, it has thymine, but RNA has uracil. That's actually really important, because they'll give you runs of a nucleic acid that'll show a 5' end and a 3' end, and it'll look something like this. So they may give you something that looks like this. And you have to be able to look at that and go, oh, okay, so you look at that with your eyeball, and you can tell that's RNA. Okay? If you had something that looked like this instead, you look at that with your same creepy eyeball, you would think, okay, we're talking about DNA, because they won't tell you. They'll just let you figure this out based on, oop, you have uracil, that means your RNA. You have thymine, that means your DNA. So this is where this particular part comes in. Excuse me. DNA is double-stranded. RNA is single-stranded. The reason is RNA tends to be used primarily to make a copy of the information of DNA. They have different sugars, so deoxyribose as a sugar, and then RNA has ribose as a sugar. They're the same sugar, except that deoxyribose is missing one oxygen. Everybody knows that DNA forms a double helix. There's no helix. RNA tends to be linear. The other part about DNA is that DNA can only be found in the nucleus. So if you have some type of dye or some type of marker that goes into and stains DNA, you'll never find it outside the nucleus. Wow. Nucleus. It's always there. And there's safeguards we'll talk about to where it can't get out, like it physically cannot get out. RNA is going to be found all over the cell. So all over the cell. And it's going to be primarily found, it goes into the nucleus, and it's going to be to make a copy of a gene on DNA. That's transcription. I think my thing's running out. And then it can easily come out of the nucleus, not a problem. So it'll come out of the nucleus and it'll move out to the cytoplasm, and it'll go to ribosomes. Ribosomes are always outside the nucleus, and it'll be used to make proteins. And that process is called translation. We have a whole unit on that. So in general, that just gives you some ideas to picture in your head. So the point of this one, there's a brain. Okay, so there's wrinkles and smart brain. So this is just so you can have a picture in your head of what this all looks like. Because if you notice, it's all about like what structure and what it looks like is. So let's just take a few more notes on DNA. So DNA, I'm just going to be able to, what if I have to talk about it? So let's put talking points, which could really be writing points if you think about it. I'd want to be sure that I said that it was only found in the nucleus. I would want to let the person know that I know that DNA contains all the information for every cell in the body. So your liver cell has the entire book of you. A skin cell actually has directions for how to make a liver cell. They're just turned off. So it contains all of the genes for every cell in the body. And I may just want to put a little note here that says all are turned off. Put the genes for the cell. We'll just put of interest. So if you're talking, they always say stuff like that, gene of interest, cell of interest. It's the one you're talking about at the time. We could put a little note here that this is done mechanically, which means we block them. They're physically blocked. So for this DNA, it's available all the time to be able to be translated. So it's, okay, so for the open, that's the part that's not been mechanically locked down. The open part of DNA, it's going to be available for the genes to be copied, and that's going to be by RNA at any time. So 24 hours a day, you're going into your DNA and you're making copies of what the instructions are for a protein, which is a gene. So for the open part of the DNA, the part that actually
codes for the proteins for the cell of interest. It's available for the genes to be copied by RNA, so that's called transcription. And this is done 24/7, nonstop. Let's just remind ourselves that genes code, I don't like that, code for proteins. So genes are in the DNA, and that's in the nucleus. So it's kind of like when you go to the library and you have a reserve section in the library and the books that are really important that they don't ever let anybody check out, you're welcome to use them while you're there, but you're not taking them out of the library. If you want any information, you'll have to copy it down. That's exactly what DNA is like. So DNA's in the nucleus. It's too important to come out. So we're going to put, like, don't you put this in a test question. So like the reserve section in the library, because it's just too important. So it's not going to do well more. Like the reserve section in the library, you can use, but you got to make a copy. So you're going to have to make a copy of the gene. That's RNA. Actually, it's messenger RNA. And then you're going to use that information. So you're going to use messenger RNA to actually make the protein. So you're going to use, let's put the information in messenger RNA to make a protein, and that's going to be at the ribosomes. I'm so sorry. The last piece is not at all the least piece. It's probably super important. It's the universal molecule DNA is, which just means life as we know it, for living things. I'm saying all living things, genetic information. Life is DNA based. That's what that means. So we could just summarize that. Life is DNA based. So if I had to talk about DNA and I wanted to show somebody, hey, I know about DNA. Like, I can talk about DNA. I want to be sure that I make the point that I understand it's always in the nucleus. It's in the nucleus because it's the most important part of a cell. It's the entire set of directions to make everything about you. So you have every gene for every cell, but each cell, obviously a liver cell doesn't need to know how to make proteins for a skin cell. So those are mechanically turned off. So if you think about it, most of DNA is off. And so that cell will only leave open the part to make its own proteins. It'll mechanically shut down everything else. And that open part is constantly being used like a little reference book to flip through, find the gene for how to make catalase. And so you'll make a copy of the information because you cannot take DNA out of the nucleus. But in order to make proteins, you have to go out of the nucleus. So we make a copy. We take RNA and we make a copy. It's called transcription. This is done nonstop. Nonstop, you're in your DNA. So you make your copy. You take that copy. That's called a messenger RNA. You take it to the ribosomes, and that is reused over and over like a copy machine to make that information, you take that information of what order the protein is going to have its amino acids. Like, what do you want to put next? Oh, I want to put glycine. Okay. It codes for the order of the amino acids in the protein. And then the final thing you want to be sure that you understand, it's part of the central dogma, which is this. I'm going to put it over here. And this is the way that information moves within the living thing. So DNA is going to contain all of it's the code for the amino acid sequence in each protein. And it's genes. So DNA is made up of genes. Genes are the code for what order you put your amino acids in for each protein. Then you go to the genes, and this information is going to be copied, because we cannot take DNA out of the nucleus, copied into a messenger RNA molecule. So you're going to copy this information into messenger RNA molecules. So then the messenger RNA is going to leave the nucleus and it's going to go to the ribosome, and we're going to use that information. This is all about information that's coded in the messenger RNA, which is just a copy of the DNA, to create proteins. So it's the directions. So it goes DNA to RNA, with genes being in the middle, to protein. That's what the central dogma is. The information moves from DNA and its genes to RNA to proteins. All right? Okay. Well, my computer didn't tell me that it was going to stop recording, so this will just be part of your notes for the nucleic acids. So anyway, we do a lot more with this later. Thank you for listening. I know this is not always the best way, but at least this is what I have to think about it. At least you have the ability to take the notes when you have time, versus you have to sit here maybe even at 7:45 in the morning while I do the exact same thing. So I think this is actually a good thing. I'm proud of you. I know it's not easy.
Sometimes it feels like DNA gets all the credit. Yes, the structure of DNA is amazing. It has a beautiful double helix structure, a twisted ladder. It has its own emoji. DNA stores genetic information and codes for your traits. However, sometimes what gets left out is how important RNA is. Without RNA, you actually couldn't get that genetic message out to your cells so that they can start producing proteins. We talk about this in protein synthesis. RNA is a very important biomolecule, just as important as DNA. In fact, RNA is even hypothesized as coming first, before DNA, in the RNA world hypothesis, something we need to make a separate video about. So let's compare and contrast DNA with RNA. First, you will find DNA and RNA in all living organisms. In eukaryotic cells, DNA tends to be found in the nucleus, while you can find RNA both inside and outside of the nucleus. Prokaryotic cells don't have a nucleus. Both DNA and RNA are nucleic acids, which are a type of biomolecule. Nucleic acids have a monomer, which, if you remember from our biomolecules video, a monomer is a building block. The monomer for nucleic acids is a nucleotide, so both DNA and RNA have nucleotides. The nucleotides of both RNA and DNA have 3 parts: a phosphate, sugar, and a base. Let's draw 6 nucleotides of DNA and 6 nucleotides of RNA. DNA is generally double-stranded, and if focusing on these 2 strands of nucleotides here, you can see they run anti-parallel to each other. RNA is generally single-stranded, so you are just seeing one strand here. The sugar in DNA is deoxyribose, and the sugar in RNA is ribose. This makes sense because DNA stands for deoxyribonucleic acid. That's helpful to know because the deoxyribose is a sugar, and nucleic acid is that type of biomolecule it is. RNA stands for ribonucleic acid, as its sugar is ribose. The bases in DNA are adenine, thymine, guanine, and cytosine. It helps to remember the popular mnemonic apples in the tree, that helps you remember that A goes with T, and car in the garage, so C goes with G, to understand how DNA bases pair. The bases in RNA are adenine, uracil, guanine, and cytosine. Notice the different one? It's uracil. So you have to change that popular mnemonic device here. Instead of apples in the tree, maybe the apples are under. Car in the garage still works though. We mentioned earlier that DNA codes for your traits, but it couldn't do that without RNA's help. In our protein synthesis video, we talk about 3 different types of RNA and their very important roles. mRNA, which stands for messenger RNA, mRNA's job is to carry a message based off of the DNA. In eukaryotic cells, DNA generally stays in the nucleus, but mRNA has the ability to leave the nucleus to take the message to a ribosome. Ribosomes make protein, and RNA is actually a major component of ribosomes. This type of RNA is called rRNA, which stands for ribosomal RNA. Finally, we discussed transfer RNA, or abbreviated tRNA. Its job is to transfer amino acids to match the correct mRNA codon. Codon charts using mRNA codons have been developed so that you can actually see which amino acid is brought for each mRNA codon. When those amino acids are joined together, they make a polypeptide chain. Proteins are made of one or more of these polypeptide chains, and proteins have tons of different roles. But we don't want to spoil it. Check out more in our protein synthesis video. Before we go, let's try a little 3-question quiz, shall we? Just pause it after the question so that you have time to think about it. Question 1. If I have 8 DNA nucleotides, how many DNA bases do I have? And also, how many base pairs? The answer: each nucleotide, regardless of whether it's a DNA or RNA nucleotide, has a phosphate, sugar, and a base. So 8 DNA nucleotides would have 8 bases. DNA bases pair like this, and that's 4 DNA base pairs. Question 2. If one strand of DNA has these bases shown here, ATTGAC, can you complete what the complementary DNA bases would be for the other DNA strand? The answer: so remember those base pairing rules for DNA and the popular mnemonic. If placing the bases in this image, reading in the direction shown on this image, they'd be TAACTG. Question 3. In a process known as transcription, a complementary RNA strand called messenger RNA has to complement the DNA. More about that in our protein synthesis video. So if I still had the original portion of DNA here, what would be the complementary RNA bases? The answer: so remember, it's asking for RNA. No thymine here, it's uracil instead. Apples under helps you remember A with U. Car in the garage helps you remember C with G. So in this portion, reading in the direction shown on this image, we'd have these RNA bases. Notice the answer is similar to the last answer, but there are uracil bases here instead of thymine. One last thing. Our models here, they're unable to show the beautiful 3D shape, the exact numbers of bases per turn, the chirality, but we've left you some links to explore because the goal of our videos is always to introduce the topic so that you can discover all the fascinating details and exceptions. Check out our further reading description to keep that curiosity going. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.